Next, we have Professor Yaakov Nachmias, who is renowned not only for his pioneering work with cultured meat, but also as the head of bioengineering at the Hebrew University. And today, he's here to share a transformative concept with us. Professor Nachmias, the stage is yours, please.
It's exciting to be here today, and part of the transition from cultivated meat to what I usually do, which is drug development, is probably going to be in the process of slaughtering some sacred cows. So I'm going to do my best to keep it exciting and to tell you what I really think about the current situation in the field and where, in my opinion, the field should be going, what we're doing about it in tissue dynamics. So when we're talking about pharma in general, we are facing a huge problem. While we have Moore's law in semiconductors manufacturing improving every year, pharma is facing Aram's law, the inverse. We're actually getting a lot worse. We are producing 15% less drugs every year, and the amount of drugs that we can make for under $1 billion of investment has dropped under one several decades ago. We're actually putting about $240 billion into research and discovery every year, while the FDA is approving only 30 drugs, which is honestly a crazy number. It costs about $2.6 billion to bring a single drug to the market. And the big vision over the last 5 to 7 years has been artificial intelligence, right? Like we heard before, this was supposed to solve our problem. And there have been a lot of companies in this space that have been trying to build generative models to try to predict which drugs are going to be better. Hundreds of millions of dollars of deals, billions of dollars of deals recently, and yet the movement in this space has been agonizingly slow. About a year ago, we had 2 drugs. Today, we have about 15 drugs in the pipeline. The first 2 have already failed. And it looks like AI capability is going to be to increase the needle by about 15%, but it's not going to change everything. Something is not working. Something is falling short, and the big question is why? Why is this happening? So there are 2 problems, and problem number 1 that we are facing is that we simply don't have enough model, don't have enough data. Large language models like Gemini, like ChatGPT have been trained not only with every single book on the internet, but with a lot of books that were not on the internet. You know, the sum total of human knowledge was poured into ChatGPT so it can make me those bullet point lists. We don't have the same amount of data in biology. We simply don't have enough papers. Gale is not big enough. We are looking at 5, 10% of the data, and we're simply not generating it at the same rate as we are generating books, newspaper articles, and opinion pieces, right? We're not writing papers in the same rate. Even if we assume that the complexity in drug development is equivalent to the complexity in language, we're talking with current rates and accounting for exponential growth, we'll be able to get there by the year 3000. So we have a problem. We do. We have a problem unless we find another way of doing it. And honestly, there are breakthroughs in this field. So one of the big ones is DeepMind. DeepMind AlphaFold has more than 90% accuracy in protein folding, mainly because it's doing something slightly different. It's not really doing generative AI. It's actually using structured data sets. You have a physical parameter space in which you work. And then you're taking the data that you have, which is limited, but it's enough to fit over that model that you know has to exist because this is the physics of amino acids. You add this data and you get very high level of accuracy, and other companies can do this just as effectively, and structured data sets scale exponentially better. This is not the only problem. The second problem is that our data, honestly, is shit. Biology is incredibly messy. Recent estimates talk about 77% of pharmaceutical studies that can't be replicated. This is data from Pfizer and Amgen. So many of the papers that we write can't be replicated. By a recent review in Nature, about 25% of papers published by the same lab can't be replicated. Now, this is not because people are cheating. It's because biology, by its nature, is noisy. We have an issue. 30% of the cell lines have been misidentified. People have been working decades on lines that they thought were liver cells and ended up to being HeLa. According to Tox21, a big EPA effort, 20% of the molecules they're getting are misproduced. And then 25% of the antibodies target the wrong protein. You combine it all together, and from 100,000 data points that you generate or that you scroll over and get, 65% of them are going to be false. Now imagine writing software when 65% of your data is wrong. What type of predictability are you going to get? And honestly, you're going to get this predictability. 70% of the drugs that work in humans don't work in rats, and about 88% of clinical studies fail. So we have a problem. So how do we solve this problem? How do we go beyond the antibodies, and how do we go beyond cell lines? Well, some of it has been presented here, and let me show you what we're doing. So the first thing we're doing is we're taking the antibodies and many of the assays out of the system. So we built the dynamics in the company that I founded, Tissue Dynamics. This machine over here, it took us about a decade and $11 million to build. It uses human organoids, so human tissues that Alan described a few minutes ago, made from human stem cells. And these tissues have sensors physically embedded in them that can measure things dynamically. So we're not relying on antibodies. We're not relying on chemicals to essentially understand what is going on inside the cells. We have sensors physically integrated in the biology. These are bionic human microtissues. And we have a lot of those tissues developed that have been heavily characterized and standardized together with our partners in ATCC. And we put a lot of time and effort to do that, developing a cardiac model that we just published in Nature Biomedical Engineering. It's the first one in the world that is multi-chambered, has an epicardium, endocardium, very, very complex, but is about the size of a third of a grain of rice. Okay? We have a brain organoid that can model Alzheimer's and autism, kidney organoid, liver organoids. These are all very, very complex human tissues. All of them are very small. And what we do is we are making them on a robotic platform. And again, you can't do this alone. And this is a partnership with Tissue Dynamics, with Renium and Beckman Coulter, allowing us to make those organoids not one or two or even hundreds, but 20 to 40,000 organoids at the same time on a robotic system like the one that Merck were now developing. And these systems can actually go into the dynamics and give you a very detailed data set that you can start relying on for AI predictions. And you get this dynamic data on how disease develops. So why do we need this structured data set? Why do we need to look at development of diseases? Why do we care about dynamics? Well, we care about dynamics because disease is a process. Okay? If you look at the example on the screen, healthy brain doesn't become an Alzheimer brain overnight. You have certain processes. You have disease that includes mitochondrial stress, metabolic dysfunction, protein accumulation, and finally you get a diseased brain. Now, most companies would look at the end point and the start and say, okay, how do we go from a diseased brain to a healthy brain? Well, we have plaque. Let's try removing it. Well, we tried that for the last 20 years. It's not very successfully. Trying to go back, trying to go to get a shortcut doesn't really work in this industry. And it's because shortcuts simply don't work in biology. However, when you have the data that tells you this is how disease progresses, you can chart a pathway that goes around, essentially goes backward, slowly going backward into, based on mechanisms and trying to unravel the disease on a fundamental basis. So this is a very different way of doing it. You can think about it as analogous to protein folding problems. Okay, so enough talk. Let's show you what actually is happening, how this can actually work. We'll start by, how do we use this data for, designing clinical studies or understanding what is happening in the clinic? So sub-cohort identification. So about 4 years ago, we developed a novel model of the human kidney, a polarized model that has the same gene expression as the human tissue, much better than most cell lines that are being used. We exposed it to a lot of drugs that are affecting the kidneys, specifically oncology medications, damage the kidney, causing kidney damage, and limiting their use. And what we found is a very specific metabolic signature and the dynamics that you see on the screen that we didn't expect. This is the signature of a fatty liver disease. But it's not of a liver, and it's not fatty liver disease. This is actually an oncology medication acting in the kidney. And it turns out that the organoids were showing us something new. They were showing us that the kidney was accumulating lipids, like the liver, in a way that was never absorbed before in mice, simply because it doesn't happen in mice. And there were only cursory examples of this happening in the clinic. Now, because we have this mechanism of actually understanding what is happening, we can try to reverse it, and in this case, the interpolation allows us to identify empagliflozin, a drug that could block the glucose accumulation that was driving this process in humans, and we can see, at least in the dish, that things indeed reversed. We could remove the lipotoxicity from the kidney model exposed to this oncology medication. So that's great. Now what? Now we have access to the amazing Israeli data centers that allow us to look at patients receiving these type of oncology medications. And we worked with Hadassah, 247 patients, multiple drugs. Here I'm showing 91 patients. And you can see that all the patients that got, in this case, cisplatin, had kidney damage and low kidney function. But the patients, the sub-cohort that got empagliflozin, that actually had diabetes, that was supposed to have more kidney damage, that sub-cohort of patients responded a lot better. They didn't have kidney damage markers, and they had much higher kidney function after the oncology treatment, allowing you to think about designing a clinical trial differently, doing exclusion and inclusion criteria differently than what you would otherwise perceive. We did the same thing during COVID, during the last pandemic, allowing us to do it not retrospectively, but prospectively. We worked with Mount Sinai Hospital to identify a novel pathway of the SARS-CoV-2 virus, specifically showing it was suppressing lipid metabolism in a novel pathway. And then our computational effort managed to show us that fenofibrate, a drug that was developed by Abbott and Teva, was able to reverse that flux, essentially limit the damage in the lungs and block virus replication. We partnered with Abbott to take it forward and design the clinical study based on our deep understanding of mechanism and kinetics of the disease, specifically realizing that we had to do this study on patients that are severe and hospitalized. Otherwise, we will not be powered to show an effect. And more than that, realizing that our mechanism was fundamentally different from the standard of care, we were able to essentially do it on top of the standard of care, which is pretty good: dexamethasone, remdesivir, and vitamin D. And the results were stunning. We saw a lack of inflammation being reduced in 48 hours, and lack of LNR activation, so there was no cytokine storm. And then the recent results that were not published yet, we see a decrease in mortality that was very significant, releasing patients about 3-4 days before the standard of care, and then significantly enhancing viral clearance. So this is going to be published in the next couple of weeks. The last thing I'll show you is something that is really hot off the plate, talking about neuroinflammation. This is a brain model. It's very, very complex. It responds to GABA. And we decided to look at multiple sclerosis. Very topical. The industry invested $9 billion in new BTK inhibitors that are supposed to block immune infiltration into the brain, but many of the clinical studies right now are crashing because of lack of efficacy. And the big question is, why is this happening? So our software was able to show that there is a good chance that the BTK inhibitors were causing damage that nobody was expecting. Over a long period of time, they were actually causing mitochondrial stress, limiting the efficacy of that medication after a month or two of treatment, something that you will simply not see in the standard assays that the industry is using, simply because they are doing very, very short assays. And indeed, the data suggested that there was not supposed to be any safety concerns, and there aren't. But looking long term, the dynamic system was able to gather 4.2 million data points and show idiosyncratic effects appearing after 400 hours of exposure. This is something that nobody else saw, and uniquely, we can modulate this. Now that we know what the mechanism is, our software can actually come and figure out which drugs would make this situation worse, lower the efficacy of your drugs, and which combinations of treatments of cohorts will make the situation a lot better. And you will be able to see an effect, and design a clinical study better, and maybe, just maybe, not fail in a phase 3 clinical study. So this is the promise, and these are the sacred cows, and thank you very much, everybody, for your time.